So these are the contents or my topic for the subject genetics. Part 1 of my topic is the regulation of gene expression. Second, the prokaryotic gene regulation. Third is the eukaryotic epigenetic gene regulation. And fourth is the transcription and post-translational genes. So part 1 of our topic is the regulation of gene expression. So in defining the term of regulation as it applies to genes, for a cell to function properly, it is necessary that proteins must be synthesized at the proper time. So tanan cells must be controlled or regulate the synthesis of proteins from information encoded in their DNA. So gene expression, the process of turning a gene to produce RNA and protein, whether in a simple unicellular organism or a complex multicellular organism. Each cell controls and when and how its genes are expressed. So for this to occur, there must be a mechanism to control when a gene is expressed to make RNA and protein. So how much protein is made and when it's time to stop making that protein because it is no longer needed. The regulation of gene expression conserves energy and space. So it conserves energy and space because it would require a significant amount of energy of an organism to express every gene at all times. So if there is more energy efficient to turn on the genes only when they are required. So um, only kanang expressing ang subset of genes in each cell makasave siya space because ang DNA must be unwound from its tightly coiled structure to kanang transcribe the DNA. So cells would also have to be kanang enormous if every protein were expressed in every cell all the time. So expression of genes, the control of gene expression is extremely complex. Malfunction in this process are detrimental to the cell and can lead to the development of many diseases, including cancer. Gene regulation makes cells different. Gene regulation is how a cell controls which genes out of the many genes in its genome are turned off or expressed. So gene regulation, um, as what we have discussed last semester, each type of cell has different set of each cell type in our body has different set of active genes. So despite the fact that almost all the cells of our body contains the exact same DNA, so these different patterns of gene expression cause various cell types to have different sets of proteins, making each cell type uniquely specialized to do its job. So, for example, one of the jobs of the liver is to remove toxic substance like alcohol from the bloodstream. So this one. To do this, liver cells express genes encoding subunits or kanang pieces of an enzyme called alcohol. So kana siya nga enzyme mag-break siya sa alcohol down into a non-toxic molecule. So the neurons is kanang um, in person's brain dili siya mag-remove of toxins from the body. So they keep these genes unexpressed or turned off. Similar sa cells sa liver na dili siya ga-send og signals using neurotransmitter. So they keep neurotransmitter genes turned off. So here. So there are many genes that are expressed differently between liver cells and neurons. So how do cells decide which genes to turn off. So I'm so in this figure there are many other genes that are expressed differently between liver cells and neurons. So or any type of two cells, or any two cells types in a multicellular organism like yourself. So how do cells decide which genes to turn on? So cells don't make decision in the sense that you or I would. Instead, they have molecular pathways that convert information, such as the binding of a chemical signal to its receptor, into a change in a gene expression. So we can also say that a cell gene expression pattern is determined by information from both inside and outside the cell. So examples of information from inside the cell is the proteins it inherited from the mother cell, whether its DNA is damaged, and how much ATP it has. So example pod for the information from outside of the cell, it is the chemical signals from other cells, mechanical signals from the extracellular matrix, and nutrient level. So as an example, we will consider how the cell responds to growth factor. So ang growth factor is a chemical signal from a neighboring cell that kanang mag-instruct as a target cell to grow and divide. So we can say that the cell notices the growth factor, and siyang mag-decide to divide. Yes, so how siyang mag-decide to divide. So this is the figure of growth factor signaling is complex and involves the activation of a variety of targets, including both transcription factors and non-transcription factors. So as what I have said kanina, ang growth factor muna siya ang chemical signal from neighboring cell na mag-instruct to target the cell to grow and divide. So this is the growth factor above, ani kani siya dari sa taas, and then this is the receptor, which is mag-divide siya, ang cell is maka-notice siya og growth factor and mag-decide to divide the cells. So part 2, the prokaryotic gene regulation. So prokaryotic organisms are single-celled organisms that lack a cell nucleus, and their DNA therefore floats freely in the cell cytoplasm. To synthesize a protein, the process of transcription and translation occur almost simultaneously when the resulting protein is no longer needed. So the transcription stops. So as the result, ang primary method to control unsa nga type of protein and pila ka protein is expressed in a prokaryotic cell. So the regulation of DNA transcription. So all steps mag-occur siya automatically pag daghag protein ang required. So more transcription occurs. Therefore, ang prokaryotic cell, the control of gene expression is mostly at the transcriptional level. So operons. Operons is a protein that are needed for specific functions are encoded together in blocks. The DNA of prokaryotes is organized into a circular chromosome supercoiled in the nucleoid region of the cell cytoplasm. For example, all of the genes needed to use lactose as an energy source are coded next to each other in the lactose or the operon. In prokaryotic cells, there are three types of regulatory molecules that can affect the expression of operons: the repressors, activators, and inducers. Repressors are proteins that suppress transcription of gene in a response to an external stimulus, whereas activators are proteins that increases the transcription of a gene in response to an external stimulus. Then finally, the inducers are small molecules that either activate or repress transcription depending on the needs of the cell and the availability of substrate. So, gene regulation in prokaryotes. In bacteria and archaea, structural proteins with related functions, such as the genes that encode the enzymes that catalyze the many steps in a single biochemical pathway, are usually encoded together within the genome in a block called an operon and are transcribed together under the control of a single promoter. These forms as a polycitronic transcript. So, in this figure, the promoter then na siya simultaneous control over the regulation of the transcription of these structural genes, because they will either all be needed at the same time or none will be needed. So in this figure, in prokaryote, structural genes of related functions are often organized together on the genome and transcribed together and under the control of a single promoter. So the operon's regulatory region includes both the promoter and the operator. So if ang repressor magbind to the operator, then ang structural genes will not be transcribed. Alternatively, ang activators may bind to the regulatory region, enhancing the transcription. So, French scientist Francois Jacob, 1920 to 2013, and Jacques Monod at the Pasteur Institute were the first to show the organization of bacterial genes into operons through their studies on lac operon of E. coli. They found that E. coli, all of the structural genes that encode enzymes needed to use lactose as an energy source lie next to each other in the lactose or lac or operon under the control of a single promoter. The lac promoter, for this work, they won the Nobel Prize in Physiology or Medicine in 1965. So, although eukaryotic genes are not organized into operons, prokaryotic operons are excellent models for learning about gene regulation generally. So, nasay some gene clusters in eukaryotic na mag-function similar to operons. So, dagan siya principles that can be applied to eukaryotic systems and contribute to our understanding of changes in gene expression in eukaryotes that can result to pathological changes such as cancer. So, each operon includes DNA sequences that influence its own transcription. These are located in a region called the regulatory region. The regulatory region includes the promoter and the region surrounding the promoter, to which transcription factors, proteins encoded by regulatory genes can bind. Transcription factors influence the binding of RNA polymerase to the promoter and allow its progression to transcribe structural genes. A repressor is a transcriptional factor that suppresses transcription of a gene in response to an external stimulus by binding to a DNA sequence within the regulatory region called the operator, which is located between the RNA polymerase binding site of the promoter and the transcriptional start site on the first structural gene. So, repressor is, as what it said, ang repressor is a binding physically blocks RNA polymerase from transcribing structural genes. Conversely, an activator is a transcription factor that increases the transcription of genes in a response to an external stimulus by facilitating RNA polymerase binding to the promoter. So, also the inducer is the third type of regulation, as what I've said ganina, is a regulatory molecule is a small molecule that either activates or repress the transcription by interacting with a repressor or an activator. So, also in prokaryotes, there are examples of operons whose genes products are required rather consistently and whose expression therefore is unregulated. Such operons are constitutively expressed, meaning they are transcribed and translated continuously to provide the cell with constant intermediate levels of the protein products. So, such genes encode enzymes involved in housekeeping functions required for cellular maintenance. So, including ang DNA replication, repair, and expression, as well as the enzymes involved in core metabolism. So, nasiya'y other prokaryotic operons that are expressed only when needed and are regulated by repressors, activators, and inducers. So again, there are three types of regulatory molecules that can affect the expression of operons. That these are the repressors, activators, and inducers. So, as what I've said, repressors are proteins that suppress transcription of a gene in response to an external stimulus. So, in other words, a repressor keeps a gene off. So, in activators, proteins that increase the transcription of a gene in response to an external stimulus. In activator, turns a gene on. So, in repressor, in other words, a repressor keeps a gene off, while the activators, it turns on a gene. So, in inducers, these are small molecules that either activate or repress transcription depending on the needs of the cell and the availability of substrate. So, inducers basically help speed up or slow down or mag-on siya or off by binding to a repressor or activator. So in other words, they don't work alone. So the third part of our topic is the eukaryotic epigenetic gene regulation. The human genome encodes over 2,000 genes. Each of the 23 pairs of human chromosome encodes thousands of genes. The DNA in the nucleus is precisely wound-folded and compacted into chromosomes so that it will fit into the nucleus. It is also organized so that the specific segments can be accessed as needed by a specific cell type. So, the first level of organization or packing is the winding of DNA strands around histone proteins. So, histone package and order in DNA into structural units called nucleosome complexes, which can control the access of the proteins of the DNA under the electron microscope. So this is the figure of DNA around histone proteins to form nucleosomes look like small beads on a string. Muna siya. These beads or kaning histone proteins can move along the string can move along the DNA and change the structure of the molecule. So, if DNA encoding a specific gene is to be transcribed into RNA, the nucleosomes surrounding that region of DNA can slide down the DNA to open that specific chromosomal region and allow for the transcriptional machinery RNA polymerase to initiate transcription. Nucleosomes can move to open the chromosome structure to expose a segment of DNA, but do so in a very controlled manner. So in figure 1, the DNA is folded around histone proteins to create a nucleosome complex. So these nucleosomes makakontrol siya og access of proteins to the underlying DNA. So pag imo siya i-viewed through electron microscope, muna siya sa B. Ang nucleosome look like beads on a string. So yeah. So this is the figure of a nucleosome that can slide along the DNA when nucleosomes are spaced closely together. Kani siya sa top. Transcription factors cannot bind and gene expression is turned off. So pag ang nucleosome are spaced apart, para sa aning nasa bottom, magka-space sila, the DNA is exposed. So ang transcription factors can bind and mag-allow ang gene expression to occur. So the modification to the histone and DNA affect nucleosome spacing. So in this top, the methylation of DNA and histone causes nucleosomes to pack tightly together, so the transcription factors cannot bind the DNA and genes are not expressed because the gene expression is turned off. So in the bottom, histone acylation results in loose packing of nucleosomes. Transcription factors can bind the DNA and genes are expressed because the genes are turned on and it has spaced apart. So the DNA molecule itself can also be modified. So this can also be modified. This occurs within a very specific region called CPG island. These are stretches with a high frequency of cytosine and guanine dinucleotide DNA pairs CG found in the promoter regions of genes. When this configuration exists, the cytosine member of the pair can be methylated, a methyl group is added. This modification changes how the DNA interacts with the proteins, including the histone proteins that controls access to the region. Highly methylated DNA regions with the acylated histones are tightly coiled and transcriptionally inactivated. So, transcriptionally inactive. So this is the figure of histone proteins and DNA nucleotides can be modified chemically. Modification affect the nucleosome spacing and gene expression. So in this type of gene regulation, it is called an epigenetic regulation. Epigenetic means around genetic. So the changes occur to the histone proteins and the DNA dili siya mag-alter og nucleotide sequence and dili siya permanent. Instead, ang changes is only temporary. Although usahay na mag-persist siya through multiple rounds of cell division and mag-alter siya in the chromosomal structure of the promoter region. So first, and mag-recruit siya og RNA polymerase to the site for transcription to be established. So this is the video.
Epigenetics refers to the heritable modifications in gene function or activity without changes in the DNA sequence. Epigenetic processes are genetic modifications that affect gene regulation by changing the DNA conformation. Epigenetic changes are found in many different cancer cell types. One such change is histone modification that may be caused by overexpression or aberrant recruitment of HDACs. Within the chromosome, DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino-terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of 2 enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made. Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones and therefore a condensed or closed chromatin structure.
So, as what I have viewed to you, this is the process of transcription in making the RNA from a DNA template. So, the last part of my topic is the transcription and post-translational genes. So, this is the a video for the transcription and post-translational genes.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the TATA box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the TATA box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA template from the strand of DNA. Most factors are released after transcription begins. When the end of the transcription unit is reached, the RNA polymerase dissociates and the newly formed strand of RNA is released.
So at what you have seen in the video, it is the transcription of the DNA in machinery. So the promoter and the transcription machinery, genes are expressed to genes are organized to make the control of gene expression easier. The promoter gene is immediately upstream of the encoding sequence. This region can be short only a few nucleotides in length for quite long, or the hundreds of nucleotides long. So the longer the promoter, the more available space for proteins to bind. This also adds more control to the transcription process. So ang length sa promoter is gene specific and can differ dramatically between genes. So the level of control of gene expression can also differ quite dramatically between genes. The the purpose of the promoter is to bind transcription factors that control the initiation of transcription. So within the promoter region, just upstream of the transcriptional starts site, resides the TATA box. This box is simply repeat of thymine and adenine dinucleotides, literally the TATA repeats. So RNA polymerase binds to the transcription of initiation complex, allowing transcription to occur to initiate transcription. A transcription factor, the TFIID, is the first box to bind the TATA box. Binding of TFID recruits other transcription factors including TFIIB, TFIIE, TFIIF, and TFIIH to the TATA box. So, once this complex assembled, ang RNA polymerase can bind to its upstream sequence. So, when magbound siya along with the transcription factors, ang RNA polymerase is kanang mag-phosphorylated. Mag-release siya og part sa protein from the DNA para mag-activate ang transcription initiation complex and ma-place ang RNA polymerase sa correct orientation na magbigin og transcription. So, ang DNA also is the binding protein na magbring as the enhancer, which can be quite distant from the gene. So in contract with transcription factors and mediator protein. So this is the figure. In this figure, as what we have the enhancer is a DNA sequence that promotes transcription. So ang each enhancer ana is made up of short DNA sequence called distal control. So distal control elements interact with mediator proteins and transcription factors. So na two different genes na may have the same promoter but different siya with control elements. So mag-enable siya to different gene expression. So in a, So when transcription factors bind to promote just upstream of encoded gene, it is referred to as cis-acting element because it is on the same chromosome just next to the gene. The region that particular transcription factor bind to is called the transcription factor binding site. Transcription factor respond to environmental stimuli that cause the proteins to find their binding sites and initiate transcription of a gene that is needed. So in addition to the transcription factors, ang other transcription factors um, can bind the promoter to regulate gene transcription. So as what they've as what is it is cited in the PowerPoint, these transcription factors bind to the promoter of a specific set of genes. So dili siya general transcription factors na magbind to every promoter complex, but mag-recruit siya to a specific sequence on the promoter of a specific genes. Enhancers and transcription. In some eukaryotic genes, there are regions that help increase or enhance transcription. These regions, called enhancer, are not necessarily close to the genes they enhance. They can be located upstream of a gene within the coding region of the gene, downstream of a gene, or maybe thousands of nucleotides away. So, enhancer regions are binding sequences or sites for transcription factors. So, pag ang DNA is magbend siya og protein, the shape of DNA changes. So, if ang shape is mag-change, it allows for the interaction of the activators na mag-bound to the enhancers with the transcription factors bound to the promoter region and the RNA polymerase. So, ang DNA is generally depicted as a straight line in 2 dimensions. It is actually a 3-dimensional object. So, ang nucleotide sequence, thousands of nucleotides away, can fold over and interact with the specific promoter. So, the post-transitional control of a gene expression. RNA is transcribed but must be processed into a mature form before translation can begin. As with the epigenetic and transcriptional stages of processing, this post-transcriptional step can also be regulated to control gene expression in the cell. If the RNA is not processed or shuttled or translated, then no protein will be synthesized. So, that would be all. Thank you.